So hello everyone. I'm happy to be here and share our vision and what we're doing at Cereno Scientific in cardiovascular disease. Our primary focus is to develop a completely new concept and a new drug for the biggest killer on the planet, thrombosis, blood clots, and I'll share some information around that with you. We are also exploring now avenues into fibrosis and inflammation. That's quite new. But let's first look at thrombosis-related diseases. So the biggest killer on the planet, and in cardiovascular disease is 50% of the deaths, and about 80% is thrombosis-related. Our approach is completely new in thrombosis. I'll share that with you. We have an existing compound that's valproic acid that's been used for epilepsy for 45 years. And so that we have reformulated that into new administration for these indications. What we have is that we have a double effect on the body's own fibrinolytic system, which we have, all of us, in our cardiovascular system, in our lungs, and so on. So it's a protective system that we have. And that's reduced, its efficacy is dramatically reduced in risk patients for thrombosis. So we have both an effect on TPA, which is the fibrinolytic substance, a protein, and we also decrease a risk factor for thrombosis pi, which is the main blocker of TPA. Now, our strategic platform continued is that we have a documented effect in vitro, in vivo, in animal studies, in experimental human studies, and in independent epidemiological studies, major studies that have indicated that this really protects from thrombosis. We also completed a phase 1 study with our drug, CS1, and it was well tolerated, had the pharmacokinetic profile that we were looking for, and also showed a significant reduction of the risk factor for thrombosis pi. And this data was released last summer. In addition, we didn't see any effect on bleedings, and I'll tell you why that's important. So now we're pursuing a phase 2 study with CS1 to document proof of concept in man that we prevent thrombosis. Now, we also have received in October last year patents for the US market, which makes up about half of the market, around $10 billion in sales annually. And in the spring, we got patents in Australia. We have extended our pipeline. We acquired a new substance from the chemists, smart chemists at Astra, that have formed a new company, Emeritus Biopharma. And these were the guys behind LSX and other compounds in Astra. LSX once was the major seller of all drugs. Now, another very exciting development that we have is that we have established a scientific advisory board of heavyweights in the cardiovascular space, and I'll share some of that work with you. So looking at the market, of course it's big because this is the biggest killer for humans. And so there are therapies out there. The older drugs, warfarin, anticoagulants, and then the newer drugs, NOACs. And you can see now that the market of drugs in this space is $24 billion a year, and it's expected to grow to $43 billion, approximately at the time when we think we'll reach the market. So it's a huge market. Why is there a need for new drugs in this space? Well, all the drugs that have been developed are weakening the coagulation system in the body. That's how they work. And the side effect of that is that you put humans at a constant risk of bleeding 24/7. And so the drugs have been developed carefully not to cause too many bleeds. And that then has a downside in efficacy. So 20% get the new thrombosis, such as an infarct, despite treatment within 5 years. So there are a double issue here on the market. You put patients at risk of bleeding, and you have insufficient preventive effect. So there is a need for new strategies in this market, and that's what we're going after. Looking at bleeds, up to 3% in clinical material get severe bleeds, such as hemorrhage in the brain. So that's 3 patients in 100. Or a hemorrhage in the stomach, etc. And up to 25% in clinical material get any kind of bleed, for instance, nosebleeds that doesn't stop. So the cardiologist and other treating physicians are worried about this, but they have no other options. What are we doing about this? Well, the body has its own balanced system of coagulation and fibrinolysis, and all the drugs that I mentioned out in the market are working on weakening the coagulation system. We are addressing the body's own fibrinolytic system, our safety system against thrombosis. And we are alone with this approach, to our knowledge. And the great thing with this is that this concept acts only locally when it's needed. So you don't put patients at risk 24/7 for bleeding. So we're helping the body dissolve the beginning thrombosis that's happening. Looking at a vessel here, you can see that's an atherosclerotic plaque. And when that sometimes and quite often actually bursts, that's an originator of a clot formation. There are signals going up to the endothelial cell wall, the inner walls of the vessels. And TPA, the protein that I mentioned, is released and dissolves the clot before it becomes occlusive. So that's how it works when it's supposed to, when it works well. Now, there is another protein circulating in the vessel and in the bloodstream called pi. And pi's main action is to block TPA. So this system is in balance. So this works very fine in healthy subjects or individuals. But when you are at risk for thrombosis, if you have diabetes, high blood pressure, previous infarcts, etc., it's out of balance, and you have a reduced TPA level in the endothelial cell wall. There's far too little in there, and you have too high levels of pi in the bloodstream. And I will illustrate that. But you might know that infarcts and stroke happens most frequently in the morning hours, and that's related also to pi. So you can see on this slide over here that pi is very high in the morning hours. That's when infarcts are most frequent, and you can see here on unstable angina and so on. And so that's when you are most at risk. Now, looking at the vessel, coronary arteries as example, you can see here a healthy patient. You get a small plaque bursting, and there is TPA being released and stops the clot from growing, and pi is in low normal levels and take care of whatever TPA is over. And then you have a patient at risk here. There's far too little of the dissolving TPA coming out, and pi is very high, especially in the morning hours, blocks whatever is coming out, and you have a high risk of occlusion. And the result is an infarct. And as you see here, if you get an infarct, the myocardial cells die. And you might die if you get ventricular fibrillation as a result of that, or you get heart failure if it's a big damage. What our drug is able to do, we're able to restore this system. So we, through an epigenetic mechanism, we upregulate the synthesis of TPA, so it's stored in sufficient quantities, and we also reduce pi significantly in the bloodstream. So we act on both these negative risk factors for thrombosis. That's what our drug can do. So we have a double mechanism of action. We increase the production, storage, and release of TPA, and we reduce TPA's blocker pi, which is high and a risk factor for thrombosis. And the beauty with this is that this is only activated by the body itself when needed, when you get a small bleed happening and a clot formation. So our objective is to develop this, of course, and with less risk of serious bleedings, and as an effective drug. And we are targeting deep venous thrombosis and primarily stroke. MI, of course, is also a possibility. But the major drug sales are in stroke prevention and atrial fibrillation. That's where the major sales are and the major need is. So we're developing a first-in-class product. that is restoring the protective system that the body has itself, and it's activated only when and where needed. Now, the data behind this concept is listed here, and I'll show you some of the data. I mentioned it briefly before. So we have documented in aortic cells that if you give our active substance, VPA, over a period, then you see here the green is TPA. This is untreated and depleted. There's no TPA here in the endothelial cell walls, and here's lots of green. And in our proof of concept in vivo in mice, where you induce a scar in the vessel, this is in vein vessels, and there are pre-treated mice and those with control. And you can see here, you can see the blood clots here. So this is the untreated, pre-treated, and this is pre-treated with our active substance. And already after 2 minutes after injury, you see there's a preventive effect from fibrogen growth. You can also see the size of the thrombosis is much less here. So we actually prevent thrombosis in live mice. Now, in man, in experimental studies, both in healthy and in coronary disease patients, we have shown that we increase the TPA release in the forearm model, and that's illustrating that there is more TPA available to release after pretreatment 4 weeks with our active substance. And 2 weeks, we have obtained up to 50% reduction of pi in the bloodstream. So both factors affected in a positive way in man. Now something very interesting, and this is work we have not commissioned or done, but there are 3 major epidemiological studies been done by epidemiological research groups in the world. The first one in Denmark, and the second one in Ireland that repeated the Danish data, and the third one came out of Scotland. The 2 first are more related into cardiovascular events such as MI and stroke a bit, the last one stroke. And they all indicate that valproic acid protects you from thrombosis and cardiovascular events, all of them. And I'll show you one piece of data here. This is the Danish study. So almost 40% less myocardial infarction. So that what they did, they went into all the people with VPA or epileptic treatment in Denmark, the whole register, and they looked at the different classes of drugs they were treated with, and they looked at cardiovascular endpoints over 10 years. And there was 40% less MIs in the group that had VPA, and nothing in the other groups. So if you were on this, you were protected for MI. And the other studies repeat basically the same. Don't have time to show it now. And then, as I mentioned, we had a positive phase 1 study coming out last summer, fall. And we showed there was no issue with safety and tolerance. We had the targeted pharmacokinetic that we wanted, which is that we want to attack the circadian peak of pi in the morning hours. So we have developed a controlled release that is a delayed immediate release to take care of that plasma peak of pi. And we showed actually in this study, which is on healthy people, a reduction of pi, even though the levels weren't that high. And we had no negative effects on bleed. So moving forward, we signed a contract with the OCT group in St. Petersburg that's going to do our phase 2 trial. And it's going to be run in approximately 30 clinics in Russia and Bulgaria. And it's going to be done in orthoplastic surgery, so total knee replacement. And there's a much higher risk of thrombosis than in normal people. So it's a good model. It's been used by Big Pharma before to show proof of concept. And then I mentioned this patent in the US. So what else has happened? Well, we established the Scientific Advisory Board, and I'll come back to that later. And I mentioned that we had acquired a new substance. With regard to the phase 2 trial, we submitted our clinical study protocols to the authorities in end of May, in Russia and Bulgaria, and we're waiting for their approval of that and expect it during the fall here. And to pursue our programs and development, we closed a share issue on 60 million in June. a 1-to-1, and we have a market cap of 170 million Swedish and about 2,000 shareholders. Now let's look at the scientific advisory board. So I've worked with a couple of these people before, very highly regarded cardiovascular researchers and professors globally. And we've been involved in survival trials and heart failure together. But this time they approached me and us at Cereno because they thought what we were doing was so interesting. And you know, we established this during the spring, and you can see our first meeting in Athens here, together, all of us. They think we should expand our focus, and the reason is that HDAC inhibition and VPA specifically has shown a number of positive effects in different organs, specifically on fibrosis. And why is that interesting? Well, fibrosis is behind a number of diseases, organ diseases and disease development in the body. And what I will just mention here is that, you know, I mentioned that we reduce pi. Pi is related to fibrosis development. So high pi levels, you get more fibrosis development. And there are a number of studies. We know that we reduce pi, and there are a number of animal studies showing that you prevent fibrosis with VPA. All right? So, of course, this adds value to cardiovascular drug. So the major cause of atrial fibrillation, where the major sales of thrombosis preventing drugs are used, atrial fibrillation, the major cause of that is fibrosis development. And I'll just show you one piece of information that was published this spring in March by the Schultz group. And I'll just mention here that when using VPA in transgenic mice prone to fibrosis development and atrial fibrillation, if you had VPA, you delayed the onset of atrial fibrillation. You prevented fibrosis development, and you even regressed it. So if you treat patients to prevent thrombosis, if you also can address the underlying disease development of fibrosis, then you have an advantage as a drug on the market. So just looking at what's been used in thrombosis here and some characteristics. So warfarin is the old drug, and you know, dosing, monitoring, so on is required. So the new drugs was developed, NOACs. You don't need to do that there. And of course, you need to have efficacy. The problem with both the old and the new, is the bleeding. So NOACs doesn't bleed less than warfarin. They bleed the same. And they have about the same efficacy, actually, but they're easier to give, NOACs. Yeah? When it comes to organ protection, there's nothing documented here. What we are offering is we have no dose monitoring. We don't expect it. We think we'll be efficacious in thrombosis. We expect to be at much less risk of bleeding, which is the major issue here. And if we can document continuously into man that we prevent fibrosis, it's going to be a major advantage. So back to the market, 24 billion, 2017, 43, and NOACs is used there, and that's the market we'll enter eventually if we succeed. If you make your estimations on, you know, price and markets, and we've done it here, of course, and this is based on taking 10% of this whole market eventually, it's $4 billion sales annually. So I'll leave it at that. Now, looking at what we're doing then, so we are pursuing a phase 2 trial here. And that should be done by the end of 2021. And then our new substance is in preclinical, and we'll return with information of that later. So what we are doing here with VPA, there is evidence of effect in thrombosis, and there is also evidence of effect on fibrosis prevention in animals. Here, it's clear. So we are now pursuing a phase 2, and we've outlined a phase 3 plan. Fibrosis is being evaluated, both in general, but also specifically for orphan drug designation. Upcoming milestones. So we are mapping the different opportunities here within coupled to fibrosis and sometimes also thrombosis. We estimate approval of the phase 2 during the fall. We're scaling up production for this rather large study this fall, and production then during the spring, and we estimate to start it during the spring, phase 2, and complete it by the end of 2021. All right, thank you.